made out of a phosphate group, highlighted in yellow, a sugar called deoxyribose, which is the blue molecule, and 4 nitrogenous bases. One of them, adenine, is shown in green. There are 3 steps involved in replicating DNA. DNA replication begins with initiation, then enters the elongation phase, and the final step is termination. During initiation, proteins bind to the origin of replication on the DNA, while the enzyme DNA helicase unwinds and opens up the DNA double helix. Finally, 2 replication forks are formed at the origin of replication, preparing the DNA for complementary base pairing. DNA helicase initiates the DNA replication process by breaking the hydrogen bonds. This process separates the double-stranded DNA into 2 single strands, in turn forming a Y shape known as the replication fork. The 2 single strands are oriented opposite of each other in order for the 2 sides to be replicated by different processes. One of the strands is called the leading strand, and it is oriented in the 3 to 5 direction. The other strand is oriented in the 5 to 3 direction, and it is called the lagging strand. After initiation, elongation begins. This begins with an enzyme called DNA polymerase, adding DNA nucleotides to the 3' end. The template strand specifies which of the 4 DNA nucleotides, adenine, thymine, cytosine, or guanine, is added. The leading strand is synthesized continuously towards the replication fork as helicase unwinds the template double-stranded DNA. The lagging strand is synthesized in the direction away from the replication fork and away from the DNA helicase unwinding. An enzyme called RNA primase generates a primer, which is a short piece of RNA, to bind to the 3' end in the leading strand, generating replication in that strand. Then, DNA polymerase adds new DNA base pairs in the 5 to 3 direction. During elongation, the leading strand is made continuously, as shown in the diagram. Due to many sites being exposed, the lagging strand is made discontinuously in pieces called Okazaki fragments. Termination begins when DNA polymerase reaches the end of the strands. Interactions between the termination site sequence in the DNA and a protein that binds to this sequence stops DNA replication. At the end of the lagging strand, when the RNA primer is removed, it is not possible for the DNA polymerase to seal the gap because there is no primer, and the progress of the DNA replication fork is blocked. The end of the parent strand where the last primer binds isn't replicated. These ends of linear chromosomal DNA consist of non-coding DNA that contains repeat sequences called telomeres. As a result, part of the telomere is removed in every cycle of DNA replication. The DNA replication is not complete until a mechanism of repair fixes possible errors caused during replication. Enzymes like nucleases remove the wrong nucleotides, and the DNA polymerase fills the gaps.